Assalamu alaikum to all viewers. I am Eman Asif, student of BS Zoology, and today I am going to discuss the topic mechanism of teratogenesis. These are the contents of this topic I am going to explain next in this video. Introduction. What is teratogenesis? Teratogenesis is a process that causes birth defects or malformations in an embryo or fetus. Teratology is the study of the causes and underlying mechanism leading to birth defects or malformations. These may include disorders without any obvious structural malformations such as intellectual disabilities. Terms related to teratogenesis. Number one is teros means monster, genesis means producing. Teratogen is an agent that can produce a permanent alteration of structure or function in an organism exposed during embryonic or fetal life. Then is teratology. It is the branch of science which study the mechanism and pattern of abnormal development. IGD Saint was first to introduce the term teratology. The next term is teratogenicity. It is the induction of malformations by teratogens. हिस्ट्री ऑफ ट्रेटोजेनेसिस 1930s में सेट ऑफ एक्सपेरिमेंट्स परफॉर्म किए गए थे जिसमें प्रेग्नेंट पिग्स को फीड करवाया गया था डाइट डेफिशिएंट इन विटामिन ए ऑल द पिगलेट्स आर प्रोड्यूस आर malformed. Then the thalidomide drug, it is a teratogen and if it is taken during pregnancy, it causes formation of the malformed embryo. Wilson's theories of teratology. In 1973, he defined teratology as a science dealing with an adverse effect of the environment on developing system, namely on germ cells, embryo, fetus, and immature individuals. He gave 6 principles of teratology. Number one is liability to teratogenesis depends on the genotype. Number two is liability to teratogenesis varies with the developmental stages at the time of exposure. Third principle is teratogenic agents act in specific ways on developing cells and tissues to initiate sequences of abnormal developmental events. Number 4 is the access of adverse influences to developing tissues depends on the nature of influence. Number 5 is the four manifestations of deviant development are death, malformation, growth retardation, and impaired function. And sixth principle is manifestation of deviant development increase in frequency as and decrease as dosage increase from no effect to the totally lethal level. Mechanism of teratogenesis. Genetic factors. Genetic mutations can result in teratogenesis. These mutations may be inherited from parents or arise during gamete formation. Mutations in genes involved in embryonic development such as those regulating cell differentiation, proliferation, and apoptosis. These can disrupt normal development of the fetus. Environmental factors. Teratogens are external agents that can interfere with normal embryonic development and cause birth defects. These include chemical teratogens, physical teratogens, and infectious agents. Chemical teratogens include some specific drugs, alcohols, tobacco, smoke, pesticides, etc. Physical teratogens include some physical factors such as radiations, UV rays, X-rays that can damage DNA and result in formation of deformed organisms. Infectious agents include some viruses such as CMV, toxoplasmosis, Zika virus, etc. These can damage developing fetus. Mechanistic pathways. Teratogens can disrupt embryonic development through several mechanisms. For example, cellular damage. Teratogens may induce DNA damage, oxidative stress, or apoptosis in developing cells, leading to abnormalities in tissue formation or organogenesis. Disruption of signaling pathway. Teratogens can also affect this pathway. For example, retinoic acid is important in specifying the anterior and posterior axis and forming the jaws and heart of the mammalian embryo, and its disruption by teratogen can lead to abnormalities. Nutritional deficiencies. Inadequate maternal nutrition, particular deficiencies in essential vitamins and minerals like folic acid, can impair fetal development. Timing of exposure. The timing of teratogen exposure during pregnancy is critical. Major structural malformations are more likely to occur during the embryonic period when organogenesis takes place, while functional abnormalities may result from teratogen exposure during later stages of fetal development. Hormonal imbalance. Teratogens can disrupt hormonal balance during critical period of fetal development, leading to abnormalities in sexual differentiation or organ development. For example, exposure to certain endocrine disrupting chemicals can interfere with normal hormone signaling. Oxidative stress. Some teratogens generate reactive oxygen species leading to oxidative stress, which can damage cellular structure and macromolecules crucial for development. Latest techniques on treatment and prevention of teratogenesis. One of the latest techniques in preventive teratogenesis is preconception counseling and screening. This involves identifying potential risks and genetic factors before conception to mitigate the chance of teratogenic effects on the fetus. Another technique is prenatal genetic testing, such as non invasive prenatal testing and whole exome sequencing, allow for early detection of genetic abnormalities, enabling early intervention and personalized treatment plans. In terms of treatment, targeted therapies and gene editing technologies like CRISPR Cas9 hold promise in correcting genetic defects that may lead to teratogenic outcomes. Thanks for watching.